All right, so our next speaker is virtual, and I'm going to echo Matilda there. We have to say good morning because he's in Sydney, Australia. It's the early morning for him. So we are happy that he's with us. So please help me welcome Dr. Thomas Ashhurst. He's a senior cytometry scientist with the Sydney Cytometry Core Research Facility and honorary research fellow in the infection disease and immunology of the University of Sydney. In this role, he specialized in the use of cutting-edge high-dimensional cytometry, single cells, and spatial multiomic technology for interrogating cellular systems. His work involved the development of new computer and experimental methods to better understand the immune development and pathology across the human lifespan and hold a long-standing interest in the pathogenesis of severe respiratory and neuroinvasive infectious diseases. All those. So welcome, Thomas, and happy to hear your talk.
Thank you so much for the kind introduction. Hopefully the audio is coming through okay. Hopefully the video is coming through okay. Excellent. Okay, yeah, it's a wonderful opportunity to share with you today, so I really appreciate the opportunity. What I wanted to speak about a little bit today is effectively a couple of challenges that we're dealing with in spatial analysis of spatial data in a cytometry context. And so, we sort of say cytometry, and generally people are thinking about like a flow cytometer or a mass cytometer, and we're sort of taking a broader view of what we count as cytometry. So this is where I work, the Sydney Cytometry Core Research Facility at the University of Sydney. And I suppose in our facility, we measure cells is our primary goal. And we do this using platforms that would be familiar with most people, so flow cytometry, et cetera. But that now sort of expands out into kind of more and more, tenuous connections, including molecular methods, including things now like spatial transcriptomics, for example, as well as high-dimensional imaging platforms. I suppose the goal of the facility is to offer this as kind of a one-stop shop for different methods of looking at cellular biology in different contexts. So that's kind of our idea. And most recently, we have created a partnership with our mass spectrometry facility here at the Charles Perkins Center to create what we call sort of a pan-omics initiative. The idea is that we kind of, we don't do sort of hardcore mass spec like the mass spec facility does. They're kind of really into like hardcore proteomics. But we share a lot of commonalities in terms of, I suppose, wanting to be able to give people an omics level view of their cells and their tissues, and potentially to be able to use these technologies sort of in combinations on different samples. And so we've kind of joined forces to create a single point of contact where people can bring, their samples and their project goals and challenges. We can sort of define a pathway through different platforms and technologies that will give them the answers that they're looking for. And so in this context, we're now, dealing a lot more with looking at multimodal data. So sometimes, we run, maybe a sample on like immunofluorescence and then a sample on, with the mass spec core. And there might be two different samples, but we're sort of getting to the point now where we can run a single sample through multiple platforms and gain different kind of, I suppose, levels of gene expression information, which can be very helpful. But it's this multimodal data that we're now having to sort of deal with a lot more and struggle with the analysis challenges around that in particular. And so I wanted to talk about kind of two challenges that we're dealing with in this context. And these are kind of almost discussions more than presentations. I think we have some interesting, I suppose, experiences in dealing with this and some interesting solutions, but I think I'm not presenting here is how you solve this problem. I think these are ongoing challenges for the field, and I'm just interested to share things that we've come across as we walk through this area, and interested to hear other people's thoughts as well. So yeah, multimodal measurement of gene expression is the first sort of challenge I'd like to talk about. And essentially the idea here is that, gene expression, so, however a gene is being expressed at a different, level through the sort of the normal dogma, in most cases for us, we're sort of looking at RNA and protein, just in a simplistic sort of sense. But we face this challenge sometimes where, gene expression does not always sort of have like a linear relationship between the two. And this is kind of captured in various studies, either looking at individual sets of samples or more sort of meta-analyses. But just because a gene is being expressed at an RNA level doesn't necessarily mean it's being expressed at a protein level. And it doesn't mean they're being expressed in a way that correlates. And this and vice versa. So this can be kind of an annoying challenge when we're using proteins as kind of, helping us identify cell types or assign sort of functional states to cells. And if we're looking at a protein-based assay, that gives us some information. But if we switch to an RNA-based assay, then potentially we're getting slightly different information even for the same genes that are supposed to be being expressed. The way I like to sort of summarize this to people when we're sort of talking shop operationally for planning is I think most genes fall in this category, which is like sort of low positive correlation. Like, yeah, if there's more RNA, there tends to be more protein, but it's not strongly correlated. And so you don't get like a nice sort of clean cut model. You get this sort of, you know, most of the genes are kind of weakly positively correlated. Some of them are very strongly positively correlated. Some sort of are anti-correlated. Some you get kind of dynamics that just on the face of it don't appear to make sense. But I think this is real, and I think this is reasonable as well. There's all kinds of reasons gene expression might be discordant at the RNA and protein level. I just listed a few here just for sort of simplicity, but some of these are fairly straightforward. Like some proteins are long-lived, and the RNA turnover necessary to maintain those proteins is very low. So by definition, you have a very strong protein expression on a cell for a particular gene, but almost no RNA. You can have things like post-translational modifications where you're getting a lot of RNA but not detecting a particular form of that protein at the very least. And sometimes this is just a dynamic sort of effect over time in terms of you get sort of RNA coming up first and then the RNA dying off as the protein comes through. So it kind of depends when in time we're looking for a particular effect. So sometimes these are technical issues, sometimes this is just biology and we sort of have to deal with it. I think I often like to make this point where, a lot of the people that we work with are, say, immunologists. They're kind of interested in like what part of the immune system is being fired up to deal with a particular problem or a particular pathogen. And so, whether we're seeing clues of that response at the RNA level or the protein level in a way doesn't necessarily matter because we're trying to look at the whole big picture of a response, not just an individual gene. But this dynamic still matters in terms of how we assign functionality to gene expression in cells. So dealing with this in sort of cellular data, like suspension cellular data, there's been a whole bunch of different work over the past, what's it now? 2026, so past sort of 10 years or so, where we can apply antibodies to cells that have oligonucleotides attached to the end of the antibody, so like a little strip of DNA, and then we can take the oligos that are attached to those antibodies and the RNA out of a cell and then measure essentially the same gene at RNA and protein expression level. There's different ways of integrating this data so you can kind of, bring different platforms together to combine information from different modalities. But even better than this is we can actually do this spatially with much greater ease, at least conceptually. So these approaches where we pull together kind of different modalities have like really intense integration and kind of analytical challenges, which is not something I'm going to talk about today. But just this is kind of, we have to infer sometimes, gene expression based on how we bring this data together. But I think spatial has this really nice thing where we can actually have the very same cell and then measure different properties of that cell using different platforms, but it's difficult to do so. One of the ways we've tried to tackle this, we've actually just put up online on this preprint here, which I've just listed on the page. So this very fortunately came online just last week. So I can just actually use this data now for the presentation. But essentially we had this kind of challenge where we wanted to be able to run large spatial transcriptomics analyses of tissues, but then follow it up with protein. A lot of the motivation here is that when we look at something like protein, sort of from a history of working in cytometry and immunology, the protein expression patterns that we look at are very, I suppose, they're very well known. When we're looking for a particular subset of cells, I have a protein expression pattern in mind. I can see it in my mind when I'm looking at the image or looking at a flow plot. I know exactly the kind of pattern I'm looking for. And so that helps me, look at a new set of data and link it to sort of decades of research that either I or the field have done leading up to our experiment. The spatial transcriptomics data is a little bit noisier and it's a bit hard to sort of see the particular pattern we're looking for. And so the goal here is we really wanted to combine these two things. So we can kind of lean on the sort of the known stability of the protein expression data, but then add in things like the spatial transcriptomics data, but it also gives us an opportunity to look at how gene expression is being regulated at different levels, in this case at RNA and protein level. So this is what the study looked like. basically we take tissue sections. These were tissue microarrays in this case. So sort of 1 millimeter cores taken out of different tissues and kind of set in a new array in a formalin block. Just for those who don't know the details, the Xenium platform from 10x Genomics, it's basically an intense in situ hybridization system. So we put probes basically. So in this case, probes for roughly up to either a couple of hundred or a few thousand gene targets. Those probes, once they ligate, it goes through this rolling circle amplification process where you basically create this big bundle of sort of, I suppose, barcoded targets that when we put the slide on the Xenium analyzer, or the sort of the imaging system, you can drop little different probes with different fluorophore combinations that will bind to those barcodes in a cyclical fashion, and you can kind of decode the image to figure out, where all of the different probes are and what probe identity they are. So this is quite a cool system. It runs, this is sort of a few days of hands-on work at the bench and then sort of a few days imaging. But the nice thing, and probably the important thing to remember here, is that this is non-destructive. So once the tissue is done with the Xenium sort of imaging, we can take it off and then do other imaging as well. And in this case, I'll sort of talk more about this later, but we can run it through a few different imaging systems. In this case, we were looking at imaging mass cytometry. This is sort of representative of a few different projects and a few different groups doing this. This is happening sort of more commonly in the facility, but this is just an example from this particular study where we used imaging mass cytometry specifically. And so just in case anyone who's not familiar, imaging mass cytometry is a fairly simple setup. We have antibodies that are conjugated to metals, lanthanide metals. They get stained onto the tissue like we would for sort of regular immunofluorescence. And then a sort of a high-powered UV laser sort of blasts off small 1 micron spot sizes of tissue. The metals that were contained in that spot fly into a cytoff, and then we can measure the abundance. And so rather than having single cell sort of measurements, in this case, we're having sort of 1 micron by 1 micron spot measurements. So this is kind of what the data looks like when we put it together. So from the Xenium, we'll have sort of an image or like a base image with our cells on it, which I've just represented in this little funny shape here. And it's kind of like an RNA-scope type data, or any other in situ probe-based data. We have a probe that will bind to its sort of targets somewhere in the tissue, and we'll get these spots all throughout the tissue. So I've just given an example of the CD3E gene here in RNA. At the RNA level. And so you just have a bunch of probes bound to wherever they happen to find their target in tissue. And we basically have this for as many different genes as we've imaged. So, from sort of gene 1 to gene 5,000. And so the data for this really is it's probe coordinates for the most part. It's less imaging data at this point. There are sort of images that are generated, but fundamentally what we get is a massive table. So we sort of have, what the probe ID was and where the X and Y coordinates of that probe were. If we then segment the data, we sort of draw a boundary around the cell and capture the transcript information per cell. We then get sort of a gene expression matrix. So, cells by genes in a big table. So that's the Xenium sort of side of it. And then when we add in the protein imaging side of it, we get a slightly different type of data. As I'm sure you know, when we do immunofluorescence or sort of IMC, we're looking at much more like of a continuum of data. So we're not picking individual points where there's a signal. We're sort of seeing that, all throughout this cell, in this little example here, there's going to be expression of the particular protein that we're looking for. And again, as with any sort of protein imaging system, we've got, as many different markers as we're imaging. It was, I think, probably 30 markers in this particular example. So we're in IMC, this is still, it's like it spots information in a way if you just think about individual pixels as spots. But basically we're creating kind of like a low resolution microscope image. So each pixel is 1 micron by 1 micron, looking at the protein expression and then their sort of pixel coordinates across the images. And again, if we take sort of a boundary around that cell, we can sort of, we can kind of take an average or a median expression for that cell. And now we have a cell sort of gene expression matrix where we've got cells by protein instead of cells by RNA. So this is quite good, but obviously there is a sort of a consistency here in terms of, the way that we take the image, the data from the Xenium stack and the IMC stack is actually quite similar. Once we have a boundary around individual cells, we can sum up the transcripts and sum up the protein expression for that same cell, which is quite important. But to do this, we need a unifying cell mask. So if we've got all this Xenium information and all this IMC information, we need a mask that is going to sit on top of, cell number 1 in the IMC data and cell number 1 in the Xenium data, such that we can kind of link them together. And this, at least conceptually, this will work really well. So we can now have the protein and the RNA for potentially the same sets of genes, but then, obviously much more information in the transcriptomic data. So this is really cool. So we don't have to do any like complicated sort of data integration to bring different modalities together for individual cells. We can just do it spatially. We know this cell is the same as this cell because they're literally the same cell. And so we just need to pull those together. Now, pulling those together, it turns out, is actually like a real hassle. So what we effectively need to do is take an image of something that is taken, like an image that is taken on the Xenium, and an image that is taken on the IMC that is measuring the same thing. And usually this would be DNA, so DAPI, or the iridium intercalator for IMC. What we need to try and do is register the two images. So we basically need, the nucleus of cell 1 to sit exactly on top of the nucleus of cell 1 from the two different platforms. So IMC nucleus sits here, Xenium nucleus sits directly on top of it, such that the images are, we'd say, registered. Sometimes we say the images are like aligned. I think sometimes that language is a bit confusing because we use alignment to mean lots of different things, depending on the context that we're discussing. So in this case, I would say we're going to call it image registration, but please forgive me if I keep referring to it as alignment. But to actually achieve this, we have to do a few different things. The images that come out of the sort of the Xenium workflow and the IMC workflow are just completely different. And we sort of need to do a bit of wrangling to get them into a format which is comparable so that we can make this process work. We have to make sure they're oriented correctly. They have to be scaled correctly in the right sort of way. Sometimes this is just for practical reasons. We sort of need the scaling to be similar. Sometimes this is because of just the software is really picky about what it's going to accept when we try and bring the images together. And then we sort of go to the registration steps where we're trying to get the images more and more sitting on top of each other. And there's a few different softwares that we can do to help us with this pathway. We basically like step along, we kind of take the images in and out of different softwares to achieve this at the moment. But where we end up here at the moment, we use a software called Visiopharm to do our actual registration. And the way this works at a conceptual level, if we have, for example, our DNA image from the Xenium on the left and a DNA image from the IMC on the right, we basically need to use what we would call like tissue fiducial markers. So if we can see a little feature on both images that is the same, I suppose, point, what we need to do is basically drop a little pin. So this little edge of the cell over here and this red pin here and here. We need to kind of tell the software like, okay, this point in the Xenium image and this point in the IMC image is the same. So they're not going to be exactly, registered, exactly aligned at first. We need to kind of say, okay, this point on the left and this point on the right they're the same point. And we need to basically cover the image in as many pins as we can, basically so that the computer knows, okay, this point is equivalent to this point between the two images. And I think talking to Emily, who is Ruth and Emily, who are two of the three lead authors of the study, I think in the end for this particular study, I think it was like 200 or so pins that were actually dropped across the entire image to make the registration work well. This kind of seems simplistic, but like when you think about, each of these imaging platforms are not just taking one single snapshot, they're sort of taking an image and then taking tiled images and stitching them together. You get all sorts of little effects where like, the cells are not going to be completely overlapped even when they're sort of fairly near each other. The more of these pins that we drop so we can teach the software to understand what points are supposed to be identical between the two, the better the process will be. But it is extremely tedious. I've done this quite a bit now, and it's an absolute time sink. It does work, but I feel like this is an area where sort of we can benefit from some speed improvements or some AI help potentially. So once we've done the pins, essentially the software will, take these two images that are not perfectly aligned, and there's a few different algorithms that can be applied. Off the top of my head, I don't know which one Visiopharm uses, but effectively we do what we call warping one of the images so that it eventually sort of matches the other. And once the warping is done, you've basically adjusted one or potentially both of the images so that now the cells sit directly on top of each other. So the Xenium and the IMC now are basically like copies of the same image, looking in this case at the DNA channel. So once we have this, once the images are aligned, we can apply that same warping to the rest of the information that we get from from the two modalities. So we can apply that to kind of the transcript coordinates, and we can apply that to the IMC sort of channels that we've measured for the protein expression. The way this works then is we can collect the data all per cell. So now the images sit directly on top of each other. We can take a segmentation sort of approach from either modality, like whichever one sort of will work better to give us our cell boundary. And then we can collect the RNA and the protein information at a per cell level, which we think is very helpful. Okay, so what actually happens after all of this nonsense? The first thing which we thought was kind of helpful is that, the spatial patterns that we see in terms of expression of either the RNA in the top row here or the protein in the sort of bottom row, the patterns are generally the same. This is not always the case, but in general, when we look for the RNA or the protein for a given gene across this entire core here, so this is sort of a 1 mil by 1 mil circular tissue core, the patterns of expression are generally consistent. And this is good. This is sort of like, okay, where there's B cell, gene being expressed, we're seeing the gene expressed at the RNA level, we're seeing it at the protein level. So that's good. So for CD20, for CD3, for KXD7, in general, the pattern is conserved. You can see there are some subtle differences here. Like sometimes you might find areas where it's more obvious in protein than it is in RNA, but in general, the pattern is preserved, which is helpful. But when we sort of zoom in a little bit, we find that the per cell dynamics are a little bit different. So we have this sort of a few examples here where we have, a cell that is going to express like a high level of the protein, the CD3 here, for example, and then have a whole lot of little transcripts here in teal. This is probably quite difficult to make out, I think, but so yellow cell here with the protein, and then these little teal dots are representing the RNA transcripts for that same gene. So CD3 in this case. So okay, that is a T cell. It's got CD3 protein, it has CD3 RNA, so that all seems fine. But there's also examples that we can find where we have protein but no RNA, and then sometimes we find transcripts that don't seem to have a partner in the protein expression. And as we look across different markers, we can see more examples of this. Sometimes we have, good correlation of protein and RNA at the single cell level. Sometimes we have only protein. We don't get a lot of examples where we see like a large amount of RNA but no protein, but there are some examples where this does happen. And if we look at the correlations, it's kind of mixed. It's a bit hard to do this at like a bulk level because it seems that, not all of the cells are going to behave in the same way, right? Some will correlate in terms of their protein and their RNA, some will not. But so the CD3 here, this is the actual sort of graph showing the correlation. Because we're actually measuring the number of transcripts per cell, it doesn't look like a normal correlation graph. You can either have 0, 1, 2, or 3, or 4, or 5 transcripts, right? So it's not a continuous variable in this case. Whereas the protein kind of is. It's strictly speaking, it's actually not continuous, but we sort of treat it as such. And so in this case, you can see as we're sort of going up in the number of transcripts we're detecting, the protein is kind of going up on average, but it does taper off at some point. Once we're up at sort of 3 or more transcripts, we've got the same amount of CD3, and that sort of largely makes sense. CD20 here, this is kind of a bit more of a straight line, like the more gene copies we're getting at the RNA level, the more protein we're seeing. So in general, this kind of makes sense, but when you look at the individual cells, it's highly variable. And I think that's an important point. And again, we sort of discussed like some of this is going to be technical, and particularly in like single cell RNA sequencing, we see this. We get effects like dropout, for example, which kind of create these technical issues that we need to deal with sometimes. But some of this is going to be biological as well, in terms of the inherent cellular dynamics. But this is cool and kind of interesting. It gives us an insight into how things are working within our tissues. But then we can apply this in our analytical approaches. And so this kind of becomes helpful because if you analyze just using RNA or you analyze using just protein, you actually do get slightly different answers. And this is maybe a little bit difficult to pick the colors apart. But so this here, second approach. So just to contextualize here, first approach is a cell typing exercise where we basically use one marker to identify each cell type. So just a canonical marker. Second approach is basically like clustering to identify like a cell type using multiple markers. So the second approach here is probably the most useful one to talk about here. And basically each time we see a little cell here, if it's blue, so there's a whole lot of cells here in blue, this is basically that cell was identified as, let's say, a T cell in this case, is identified both by IMC and by Xenium separately. So if you cluster by protein or if you cluster by RNA, you still find those same cells that you can identify as are identified as a T cell. But you do get some cells where you see you can only get the identity by like basically Xenium picks it up or IMC picks it up, and you get these sort of differences. They'll see a cell which is like, well, that's a T cell according to IMC because it has CD3 protein, or it's a T cell according to RNA, but not at the same time. So this is kind of these examples where this would be a cell which is identified as a T cell in IMC, but Xenium wouldn't probably pick that up because it's not seeing the CD3 gene, whereas this cell would be identified by both platforms. And so what you can see is like there is kind of variation across here. Quite a few cells will be identified the same way by both, but there are obviously quite a few through here. It's maybe a little difficult to see, but there are many that are identified by one platform only. And so this kind of creates a bit of like a conceptual challenge. Like, well, how do we know if we're running a Xenium experiment and we label something as a T cell? Like, how do we really know that it's a T cell? Maybe it's not. Maybe it's just missing that RNA. We can use a few other tricks because we're doing such much larger analyses using Xenium. We've got a few thousand genes that we can look at. We're not just relying on individual canonical markers. We can sort of pull in, CD3, CD2, etc., to get a more consistent identity across genes, which is important. But I think there is also just a reality where like we tend to look for our canonical markers. Like we look for a CD3 expression, whether it be RNA or protein, to kind of give us our final answer in a way. And like I don't think there's anything like wrong with that per se, but it does kind of reveal some limitations to how we're doing cell type identification or even sort of functional attribution if we're only looking at one modality. So I think this is kind of interesting. This gives us a bit of insight into, we're getting slightly different answers depending on how we're approaching our data sets in this case. That I think is inherent to any kind of particularly advanced platform and any sort of messy biological system. So I don't think there's anything wrong with it, but we do need to be aware of it in terms of how we, I suppose, assign value to the information we derive from these different platforms. There's sort of more to this story in terms of some kind of interesting basically tumor biology in this case, about like how these dynamics look in different sort of sections of the tumor. This I won't go into in a huge amount of detail, but the lesson is effectively the same. Like depending on how we look at this data and how much kind of trust we put into each modality, we do get slightly different answers about what we find. So this I think is just kind of important to think about. So this is kind of an interesting, I suppose, technical insight into how some of these platforms work. We're having to deal a lot more with spatial transcriptomics data and kind of multiplex immunofluorescence data and trying to figure out, like which do we use? How often do we need to use both? Is this something we need to do routinely? These are all kind of important questions for us at the moment. And to that effect, in the facility, this is less on the analysis side and more on the sort of operational side, but we're trying to make multimodal workflows as standardized sort of as we can in our sort of day-to-day operations. So that way these choices are easy. I think one of the biggest, let's say, barriers to actually doing this in the real world, kind of in a day-to-day sense, is kind of, it's like the logistics. It's sort of, if you're going to take something on Xenium and run it on a different platform afterwards, like what needs to happen? What pre-planning do we need in place before we can do that? As a simple example with the Xenium, if we use one of the cell segmentation kits that comes with the Xenium, so it stains the tissue for a few different proteins to help with the segmentation. That's really helpful, but these are all, I think, like rabbit sort of host antibodies, so like a rabbit anti-target, and they are somehow adhered to the tissue very firmly. The Comet, for example, it uses sort of a primary purified and then a secondary labeling approach. And so any secondary anti-rabbit is going to have some cross-reactivity with the antibodies that are stuck on the Xenium slide. There are ways around this, so it's not like an unsolvable problem, but it's just like there are some logistics there that we need to have pre-planned as part of our workflow. So when we're sort of sitting down with users to plan out their project, if we're going to do a Xenium and then a Comet, there's a few things we need to figure out before we start, executing the assay. Each of these different platforms we've been running in the facility. So the PhenoCycler here is part of the Melanoma Institute. So it's not our platform, but it's sort of users come to us for Xenium and then go to the PhenoCycler. We have all of these working at the moment, which is pretty cool. And each have kind of their unique challenges in terms of getting them to work. Obviously, there's one factor here. So if we go something like Xenium and then Comet, both of these are non-destructive approaches. So we could go from Comet to something else. The Comet also elutes whatever antibodies we applied to it as part of the staining. And so now we've got a relatively unstained section apart from the segmentation antibodies from Xenium. The phenocycler-fusion, the antibodies are sort of permanently stuck. It's really the fluorophores that get swapped out. And so that is still, it's not necessarily destructive. There are some limitations to what we can do with it after the fact. And imaging mass spectrometry is obviously a destructive technique. So whatever we want to do in terms of our imaging, even if we went something like Xenium Comet imaging mass spectrometer, IMC would have to be last because it sort of destroys the tissue as part of its imaging process. So yeah, operationally, there's some things to think about, which is, kind of irritating us on a daily basis, but it's a good challenge to have, I think. Okay, so the second challenge, which I want to talk about, is cell segmentation. So I alluded to a point before where we have effectively, I suppose how we describe it is, if we can have a cell segmentation mask that works well for Xenium and for IMC, we can kind of pull all the data together and we get all these unique insights. But like cell segmentation is difficult. In some cases, it can be quite simplistic. In some cases, it can be extremely irritating to get to work properly. And just to kind of, if we get one of these sort of pretty images, this is an image generated on our Comet platform. It's like, it's really cool and it's really insightful. We can look at some really interesting stuff, but we have this problem where, okay, we need to segment this. We need to kind of take the individual cells and get a boundary around them such that we can say, okay, this cell is a T cell, this cell is an endothelial cell, etc. And sometimes this can be simple, and yeah, sometimes this can be a bit difficult to figure out how you would even approach this, algorithmically or even manually sometimes. So in our case, the normal segmentation approach, which is kind of one that we've used with IMC data, particularly for the last several years, based on approaches from Bodenmiller's lab, we take a raw sort of image, so like some kind of channel data from the IMC, so DNA and maybe some surface markers, depending on what's going on. The typical workflow looks a little bit like this. We need to identify the nucleus. This is how most segmentation approaches work. Not all of them, but most of them. We sort of identify like the anchor for the cell, kind of the center, really. And usually that's identifying a nucleus based on either DAPI for immunofluorescence or something like the iridium intercalator for imaging mass spectrometry. We then need to kind of identify where the cytoplasm or where the sort of the body of each cell is, and basically figure out like where the boundary between them is. And once we have sort of a ring around the cell, then that's kind of our boundary. Anything that's inside that ring, we can count as part of that individual cell. But there are a number of situations where this can be quite challenging to implement. The first and kind of most obvious for the nuclear-based approaches is like when you have non-nucleated cells. Sometimes we have erythrocytes. This is from sort of a mouse model of encephalitis, where we have extravasation of red cells, so literally hemorrhage into the space, and actually being able to kind of count the red cell extravasation into the parenchyma here is actually quite helpful, but most of our segmentation approaches just don't pick this up because they're anucleated. Sometimes we get things like the DNA is just really fuzzy. This happens on IMC in sort of mouse lymph tissue for fresh frozen sections quite a bit, where you don't get clean like nuclei. You just get these kind of fuzzy, balls that sort of sit proximal to each other. This is really annoying to try and separate in terms of identifying the nuclei. Some cells are kind of non-uniform or just a bit weird. I really like astrocytes as an example of this. Astrocytes have these weird shapes, and sometimes they're just missing a nucleus, not because they're anucleated, but because the nucleus is sort of not included in the section. Like you take a 5-micron section where you cut through an astrocyte and you've collected a whole bunch of the cell body, but missed the nucleus. And this happens actually more than even I would have thought. And sometimes this is a problem where we just have direct overlap. The T cell and the B cell are sitting over each other in such a way that you've got, what is, when you sort of sum all the expression information together, is a co-expression event. And then in some tissues, we have this all happening at the same time, which is especially irritating. So the overlap of cells sometimes, sometimes we think about this in like a simplistic way, like maybe this is just a resolution problem. So like with IMC, for example, if we had 2 cells just here in this example sitting next to each other, if we're sort of taking 1 micron, like ablation sizes, so like we've got a 1 micron by 1 micron pixel effectively, any pixel that contains a little bit of the cell on the left and a little bit of the cell on the right is going to create, like those pixels are going to be a combination of, let's say, CD3 and CD20 in this case. And so depending on where the cell boundary is drawn, either the T cell is gaining a bunch of the CD20 or vice versa. And this could just be a resolution problem. Sometimes this is like a sample, like a literal sample problem. Sometimes the cells are sitting on top of each other, and what is effectively happening if you kind of turn the slide on its side is like the cells are literally on top of each other, but because we're doing a two-dimensional imaging sort of approach, like from a top down, we actually can't really differentiate the two. So they're not physically, sort of inside each other in the way that it looks in a 2D sense, but like they may as well be for sort of analysis purposes. Some platforms, we actually can do a level of Z stacking, but not all of them. So this creates just kind of a practical problem that we need to deal with a little bit. So an approach that we came up with a little while ago, which I'll just share briefly now, just to sort of bring this to a close, we created an approach which uses the multi-cut module within the Elastic program to create a boundary-based segmentation, and then we sort of pull the data into R to analyze it using our sort of toolkit, which we call Spectre. And essentially, it works like this. You can take sort of the raw pixel data for DNA and any one of a number of cytoplasm markers. We draw, like physically, almost like Microsoft Paint, we draw across the pixels that we're trying to train this machine learning classifier to see. And we're teaching it to see the cell body, the boundary of each cell, and then like the background signal. And then we're essentially asking it to predict for each pixel what it thinks it belongs to. Now, this looks quite similar to the sort of the nuclear-based approach I talked about before, but it's distinct, which I'll come back to in just a second. We essentially then go through a process where we kind of convert it into different formats until we get a cell mask, a polygon that sits on top of the cell that tells us where the boundary is. Now, what we can do with this approach is we're not actually saying, okay, find the nucleus and then sort of figure out where the cytoplasm is. We're just teaching it to see the boundary. So anything where we can draw a line around the boundary of a cell, and then we can tell it, okay, here's inside the cell, here's outside the cell, we can teach it to recognize that pattern. So this is the astrocyte example I brought up before. If we can just draw a line around the boundary of that astrocyte and then say, okay, yellow here is the inside of the cell, red here is the outside, we can teach it to see that same pattern across the image. Regardless of whether it's nucleated, regardless of whether it's kind of a complex shape, as long as we can draw some kind of boundary, then this will work. And we can also do this for different cell types at the same time. We can sort of take multiple layers of information to do this training. So it can see, in this case, boundaries of, in this case, microglia and, astrocytes and microglia. And it doesn't require like a pan-cellular marker. So as long as there's some positive marker, then we can kind of make this work. And then in particular, what we can do here is we can do different layers for different types of cells. So let's say we just decide, look, the astrocytes and the immune cells are two kind of different. It's a bit annoying to do them as one layer. We essentially treat them as like different things in the image. So we can sort of have a unique masking approach for the astrocytes, and then for the immune cells, they each get their sort of, fit for purpose, boundary-based assignments. And then we can bring that back together. So we've got a single set of masks that will cover each of them. It's an approach which is like, it's doing this is quite manual. Even though it's a machine learning approach, you still have to kind of go and click and do the training. We're not doing any sort of pre-trained models that we're leaning on. This is all, you come with your weird image, which is not, playing by the normal rules. But as long as you can see a boundary, you can teach the computer to see what you're seeing. And then essentially what happens is we just pull it through this approach where we say, okay, we're going to take the cell masks. We're then going to look at, what all the different expressions are. We take like what regions the cells are in, and then we're basically doing clustering and sort of neighborhood analysis as we would for any other platform. This gives us kind of quite a lot of information. We can look at the cell phenotypes. We can look at kind of what the neighborhood compositions are like. Sorry, my office lights just turned off of their own accord. This is quite a powerful analysis in our view, because we can kind of teach the computer to see anything that we can see in terms of where these cell boundaries are. Okay, so what I've shared with you today is hopefully some interesting thoughts, but probably more problems than solutions. Largely to do with how we're trying to do multimodal measurements in cells, and kind of how important it is to be looking at different levels of gene expression depending on what our questions are. Some of this is an analytical challenge, but a lot of this is kind of a more technical challenge. Sometimes this is software in terms of getting the bits to work. Sometimes this is planning for making sure these assays are going to work correctly. And then also there's issues to do with segmentation and trying to have, I suppose, as many different options as we need, such that we can segment the images and the kind of the cell types that come through. For researchers who are working on a very particular type of tissue or particular cell type, I suppose once you've found your method that works, you can kind of stick with it. We certainly find in the core, like we get all sorts of cell types, all sorts of tissues, all sorts of qualities. So having sort of a toolbox of different approaches that we can test in sort of rapid succession seems to be key to us being able to get our job done properly and support our users as best we can. Okay, so those are my thoughts on this topic. I hope you found it at least a little bit interesting. Just a couple of key people to point out here. So our core facility directors and Jason, who is our spatial biology specialist, who's sort of a powerhouse of Comet, the Comet in particular, but also our spatial analysis. Felix and Giovanna are my co-developers for the Spectre package that I mentioned. And the list here, these are sort of the key players in the multimodal study that I just described. So happy to take any questions and thank you for your time.
Thank you very much for a great discussion on that. If anybody has any questions, they can head to the microphone. But if not, I will get started with something maybe broad and conceptual, which is how do you feel that people like us who are with more of the cellular or protein background can interface with people with genomics background to kind of figure out how to do these integrations maybe more efficiently? Is there a platform for that, you know? Because I hear kind of something similar, which is the people with genomic backgrounds are developing their own algorithms for data integration, and they're all sort of individually pieced right now.
Mm-hmm. I think that is right. And we certainly have that experience here. So I mean, my background is sort of flow immunology, histology, not genomics. But moving more to that space and moving more to talking with the bioinformaticians, there are things that we take for granted sometimes, like trying to explain to the genomics bioinformaticians what the tissue artifacts are, like the staining artifacts, and they're like, well, there's signal here, and I'm like, yeah, but it's not real signal, and they're sort of like, well, how do you know? And it's like, well, we just know, it's just intuition, which is not there's not an evidence-based answer. I don't like that. I think my experience from like seeing this in multiple labs and multiple kind of institutes now is that the way this works best is when those disparate teams spend a lot of time together. I think, out of any like way of dealing with this or any like software package I've seen to deal with this, if the people spend more time talking to each other, even just like sitting near each other, like in Yvonne's size lab in Belgium, they just have the wet and dry lab people sort of sitting next to each other, and they just, I don't know, it's almost like osmosis. They start to understand more of each other's worlds by spending time together. We're lucky that we have kind of an integrated team which facilitates that now, which is really helpful for us. But I think I'm not sure there's one solution, but I do know the underpinning solution is the right people having to spend time with each other to understand more of each other's worlds. That's the one message that I suppose I'd put for that question.
Thank you. We've got one from the audience. Oh no. In one moment. Or you can come here, Ray, if you want. It might be dead. Do you want to just come?
Hello, Thomas. It's Rui here. Nice to see you.
Nice to see you.
Really great talk. Really amazing tools that I mean, as usual, you guys have been developing. I was just wondering this last approach with the segmentation. I mean, how do you? I think you touched a little bit on it. How do you guys validate or really make sure that you are really quantifying? I mean, based on the high throughput that this approach is. I mean, how do you know that you are really quantifying things properly?
Yeah, that's a great question. I'd love to say there was like an algorithmic or like a metrics approach to that. Well, no, that's not true. There are metrics for this. I look at every image that has a segment like that has masks put on it, and I check. It seems really silly, but I guess I'm looking at an image and I'm interpreting what I consider to be a cell based on experience. So human learning, not machine learning. So I suppose like if I had to draw each individual mask, it would take an eternity to do it. But I can get the computer to run it, and then I can look at the outputs and I can say like, yeah, it's just not cutting it. And so like let's refine the training a little bit, do a new output. Or yeah, that's better, but it's not quite there. So it is manual in that sense. I think it's relying on the experience of people who know the biology or know the tissue. And maybe that's not the best way it should be done, but part of me thinks it is the best way it should be done. Like I have to look at it and I have to go, yeah, I think that passes muster, or I think it's not good enough. some of the approaches that we've used before are not good at that. They sort of, it's quite, the outputs are not easy to interpret or sort of review rapidly. So part of when we did this in Spectre, the idea was like that like yellow wireframe of where all the masks are gets outputted immediately. So you can look at it, you can be like, yes, that's good, or no, let's keep going. So I think there is a lot of experience-based validation for these, and part of me just thinks it has to be how it's done.
Okay, thank you.
Okay, it looks like no more questions for this talk. So let's all thank Thomas again for a great talk. And yes, and all the way from us, waking up in the morning to do this for us. So thank you, Thomas.
Thank you so much. Appreciate it.